All right, we're going to start our lecture on the cytoskeleton and cell movement. Chapter 13, I did clean up some of the slides, but obviously the one that's uploaded is the right presentation here. And remember, after you listen to the lecture, there will be a quiz that you need to do as well, okay? All right, so first, think about, we all know what the cytoskeleton is. We've talked about cytoskeleton briefly, in previous courses and maybe even in this course as well. So we're going to kind of just talk about the main components, but then we're going to break down each component in more detail. Okay, so we're going to talk about actin filaments, talk about myosin and myosin motors, we'll talk about microtubules and microtubule motors, meaning the ability of things to move around on those microtubules, and then intermediate filaments. Okay, so we think about the cytoskeleton, we always think of it as something or an organelle that provides structure to the cell, and that's true, right? It extends throughout the cytoplasm. It also helps determine cell shape, and we're going to talk about that. It also positions organelles. It also moves things around, like I mentioned on the last slide. And then, again, just kind of provides not only structure, but organization to the cytoplasm, and again, where things are located in the cell. Okay, so it's also responsible for not only movement inside the cell, right, which it kind of says there, and I mentioned on the last slide, but also movement of entire cells. So the cytoskeleton is actually involved in migration of cells and the movement of that entire cell. And so the other thing to think about is that it's totally a dynamic structure, okay? So it can basically reorganize and itself based on whatever's going on in the cell. So again, think about a cell that's migrating. It needs to change shape. Well, that cytoskeleton can sort of polymerize or increase, and then it can kind of depolymerize or actually get smaller, okay? So it's continually changing, okay? So it's not this sort of rigid structure that just provides, not to use the same word again, provide structure to the cell. It's totally dynamic, and it's involved in a lot of different processes. Okay, so we definitely should know this. The cytoskeleton, I'm sure I'm going to ask you this somewhere on the quiz or on the exam or both, that the cytoskeleton is composed of three main types of proteins, okay? Those are actin filaments, we've all heard of all these, microtubules, and intermediate filaments, okay? So those are the three main components of the cytoskeleton, and we're going to talk about each one of them in detail. Okay, so the first one we'll talk about is actin. Right, if you look at the title on the top there, so structure and organization of actin filaments, right? So what makes up the actin filaments? How are they? What's their structure and how are they formed? Okay, so it says actin polymerizes to form actin filaments, right? So again, remember, actin's a protein. Okay, it's going to sort of join together, and we're going to see what this looks like to form these filaments, okay? And sometimes you'll see them called microfilaments. They're about 7 nanometers in diameter, so really small, okay, and about, a couple microns in length. Okay, so they get organized also kind of even further into these sort of bundles and these 3D networks. Okay, so if you just look at a picture here, right, this is an electron micrograph just showing you these actin filaments, okay? And so again, remember, they're made up of actin monomers, right? Single actin proteins that join together to form these filaments, okay? And then these filaments will form other types of structures as well. Okay, now, in order for those other types of structures to form, right, like these networks and these bundles, you have to have other proteins that help out. So we said actin itself is a protein, right? There are also actin binding proteins, okay? So these guys will regulate assembly and disassembly of actin filaments. Like I said, they'll help form these bundles and these networks, and then they'll help also make associations with other cell structures, right? Again, the cytoskeleton does provide structure and position of organelles, so it has to have the ability to bind, like, to the plasma membrane, right? We already talked about it binding via other proteins to the nuclear lamina, right? And so it has to be able to do that, and also, to the extracellular matrix. Okay, so there's all these actin binding proteins that help sort of link these together. Okay. All right. Again, I mentioned this too, that you have this. It's a dynamic structure, right? That you have polymerization, and also you have depolymerization, meaning these actin filaments can increase in size, and then they can also decrease in size, okay? And this is super important for cell movement and changes in cell shape, okay? All right, and so it's just saying that these are controlled by signaling mechanisms, right? So bottom line is this: there's going to be something that triggers a cell to move, right? Or triggers a cell to change shape. And so there are essentially receptors and other proteins that would trigger these responses, okay? So here are some examples of actin binding proteins. Again, you're not responsible for memorizing these. We're going to talk about some of these, but the point here is that there are many, and there are many others, okay? Even there are many just listed here, and you can kind of see how they have alternate functions here, depending on what you're talking about. So again, we're going to talk about some of these and the function of some of these. You are not responsible for this table, but responsible for some of the other ones that we talk about, okay? And I'll kind of tell you that as we go along. All right, so a little bit more history about actin. Or again, we're going to talk about actually how it's formed, and some of those the roles of some of those other actin binding proteins. So 1942 was first isolated from muscle cells. There's a lot of it, okay? So it says about 5 to 10% of total protein is actin in all eukaryotic cells. So if you think about that, that's a pretty high level for one protein. Okay, and there are multiple genes that will code for actin, okay? So you'll have specific actins that are expressed in muscle cells, right? And some that are expressed in non-muscle cells, because obviously it has a role in all cells. Okay, so very similar, right? So if you look at yeast actin, it's about 90% identical to mammalian actin, okay? And also some of those actin-related proteins, or proteins that look like actin, you'll see them in bacteria as well, okay? So this sort of function of actin and actins themselves are highly conserved across all organisms. Okay, now this is talking about, okay, what happens when these actin monomers, right, join together to form these filaments and then form some of these other, we're going to talk about some of these other structures, okay? And so this was only determined in about 1990 in terms of how this actually happens. So again, actin monomer, right? You can call it globular actin. That's not really a term that you need to pay too much attention to. But there are binding sites on each actin monomer, and basically that's going to help form these filaments, okay? So if you see globular actin, that's just talking about one actin monomer, and you're going to see a picture in a second, it'll make a little more sense. As opposed to filamentous actin, meaning there are a bunch of these actin monomers together to form those filaments, like I showed you in that other picture before. Okay, so this is showing you here, right? Here's your actin monomer. What will first happen is they'll form dimers, okay, then trimers, and then those trimers will join together to essentially form that filament, okay? And so that's what this F-actin is, right? G-actin globular, F-actin filamentous, okay? And note that you have sort of two different ends. There's two sort of distinct ends, and I'm going to show you why that's important in a second. So there's something called a pointed end and a barbed end. All right, you don't need to worry about this too much, but this is showing you this ATP. I'm going to show you a different picture that's going to make more sense, so don't worry about this picture too much. Just, again, showing you the filamentous actin in another way. Okay, so I just showed you that if you looked at that actin filament, that there was a pointed end and a barbed end, right? So that means it has polarity, meaning the two ends are different, okay? And so that's what this is telling you here. So it says it's important in their assembly, right? And also in establishing the direction of myosin movement relative to actin. And so myosin and actin interacting with each other is super important in muscle contraction, okay? And we'll talk about that. Okay, so now again, a little bit more about actin polymerization, okay? And again, this pointed end and this barbed end. All right, so let's look at the picture. This doesn't really say that much. We're going to look at the picture and talk about something called treadmilling, okay? So it says the barbed end of the filament will grow 5 to 10 times faster than the pointed end. Okay, so if you're talking about actin filaments increasing in size or growing, that's going to happen at the barbed end, okay? And so at the barbed end, you have actin bound to ATP, okay? And then it's then hydrolyzed to ADP, okay? So ATP and ADP play a really huge role in the polymerization and of actin, okay? All right, so if actin is bound to ADP, okay, it's going to be less tightly bound than if it's bound to ATP. Okay, and so that's why it dissociates at the pointed end, right? So remember, I just told you at the barbed end, that's going to grow a lot faster. Those actin filaments, right, are going to be able to be bound together there, okay? However, if you have that actin bound to ADP, it's less tightly bound, and essentially those actin filaments will fall off, okay? Okay, and so again, this is called treadmilling. So let's look at a picture and you'll see why it's called treadmilling, okay? And this all kind of makes sense. So really all of the, the last 5 slides that I was just describing is really pertaining to this picture here, okay? So if you look at the title of the figure, treadmilling and the role of ATP in actin filament polymerization. So if we're talking about polymerization, right, it means we're adding, okay, actin, essentially actin monomers, right? But really they're those trimers that we saw, but you're essentially growing the actin filament, okay? So if you look here, here's the barbed end, okay? You can see here that these, essentially, if it's gonna grow, it's growing on this end, okay? Because these guys are bound to ATP, okay? And therefore they're tightly bound to each other. As it sort of moves down and as you sort of add here, once they become ADP bound, they're gonna be more likely to fall off here, okay? And so again, this is sort of this treadmilling where that you're kind of growing on this end, you're kind of a actin bound to ADP, right? Again, will have the tendency to fall off. If it's bound to ATP, it's going to have a tendency to stick together with its other, actin partners. Okay, and so this is called treadmilling. Again, you're going to have growth at the barbed end, not at the pointed end. Okay. All right, now there are many drugs, okay, that will affect actin polymerization. Okay. And so they're used, even some of them are used for treatments, and we're going to talk about that. But they're also used in the lab, right, to basically disrupt function of actin polymerization to see what effect it might have on the cell. Okay, and so here are some examples. Cytoskeleton, right? Cytoskeleton will bind to the barbed ends, and they'll basically block elongation. Okay, so these drugs will bind to that barbed end, which is over here, right? And if something is bound here, preventing these guys from being added, no polymerization, okay? So it's going to stop the growth of those actin filaments, essentially, okay? So it says this can inhibit movements like cell division, okay? So again, if the actin cytoskeleton or the actin filaments aren't able to grow, it's going to inhibit all those functions that we talked about before in the beginning of the lecture. All right, so another drug here, phalloidin, will actually bind to actin filaments and prevent dissociation, okay? So here, right, it'll bind here and it'll basically prevent these guys from falling off, okay? So it says it can be labeled with fluorescent dye to visualize actin filaments, right? So again, these are kind of things that are used in the lab, right? So if you have that phalloidin bound with a fluorescent dye and you allow it to bind to the actin, it's just going to stay bound, right? And then you'll be able to see those actin filaments, okay? So, I mean, I guess you can kind of think of it as sort of stabilizing the actin filaments. Okay, now I mentioned this before too. So sure, you have those actin filaments that form, okay? So that's what we just talked about. We talked about the process of forming actin filaments and it's treadmilling, okay? So again, if it's gonna grow, it grows on the barbed end, okay? And on the other end, on the pointed end, those actin molecules have a tendency to fall off, okay? Those filaments though, again, we're thinking about, forming the cytoskeleton, and it's pretty complex. And, there's various functions, right? So these actin filaments will form different structures, okay? Again, depending on where they're located in the cell and what their function is. So two examples of how they're organized. So actin bundles, okay? So I'll show you a picture. So they're cross-linked into sort of parallel arrays. And then these actin networks sort of form these 3D, this 3D mesh, okay? And so if you look here, this is showing you a bundle, okay? So you can kind of see that it's kind of like, these are just the actin filaments here and sort of in parallel, right? And then these little purple guys are proteins, right? So they're actin binding proteins that basically help form this sort of secondary structure, right? If these proteins weren't here, then the filaments would not be attached to each other or basically arranged in this bundle. If you look here in this network, this is more mesh-like and more 3D-like, right? Where you're going to see there's other types of proteins involved. Again, this is another actin-binding protein that's going to be involved in forming these networks, okay? So this is definitely a little more complex, okay? And so you can see here some of these bundles and also the network there, an electron micrograph, okay? Let me just double check that that's definitely, double check the picture there. Yeah. Okay, so, right, like I said, so some bundles here and this network. Okay, now, when you, we said that the cytoskeleton, right, in general, the cell cytoskeleton, the organelle, is composed of actin, microtubules, and intermediate filaments. We're still talking about actin, okay, and a lot of what, we're still going to talk a little bit more about actin. Each one of those components of the cytoskeleton has a slightly sort of different role, okay? And so now we're going to talk a little bit more about function here of actin and kind of where they're concentrated, right? So it says they're concentrated at the cell periphery because remember we said it's important in changing cell shape and also in cell movement, right? So if you think about a white blood cell, right, that's going to phagocytose a bacteria or something like that, or right? It needs to be able to extend, that cell has to extend out and engulf, and that's really due to the actin cytoskeleton, that it's able to change that shape, okay? So these are some of the things we're going to talk about. That's just one example. It'll come up again. Okay, so it says this network and associated proteins called the cell cortex, okay? Again, we talked about this, determine shapes, and it's involved in stuff like movement, okay? So all this means, when you think about cortex, it's like the outside, right? And so, or the periphery. So this means that these are sort of actin filaments or this actin cytoskeleton that's right at the edge of the cell, okay, close to the cell membrane. Now, a lot of studies have been done using red blood cells, okay? If you're looking at the cortical, so that refers to the cytoskeleton by the edge of the cell or closest to the plasma membrane, right? Because red blood cells are basically just membrane. And then, you know, there's no organelles, there's not really any nucleus. It's really just cytoskeleton, membrane, okay? That's pretty much, and then obviously the associated proteins. So they're used a lot to study what's happening in that cortical cytoskeleton or the actin cytoskeleton that's right near the edge of the plasma membrane, okay? Right, so again, it's really just this actin cytoskeleton there. Okay, so if you look at these red blood cells, right, they're these disc shaped red blood cells, and again, there's not much to them except the membrane, actin cytoskeleton, right? That's pretty much what they're composed of, okay? And again, associated proteins. So here is one of these actin binding proteins. So I told you we'll talk a little bit about some specific actin binding proteins, and it's called spectrin, okay? And so it's a member of a family of actin binding proteins called the calponin family. Spectrin is just one member of them, okay? Again, I'm not going to ask you about spectrin's structure, but it does actually, you know, sort of make sense or kind of help with the function, right? So that's why we always sort of mention structure, right? Because it always correlates with or corresponds to the function of the molecule. Okay, so again, no, I'm not going to ask you about the exact structure of spectrin. Okay, but if you look here, this is showing you that structure, okay? Again, not going to ask you to draw it, not going to ask you how many beta chains or how many alpha chains are in there, okay? But again, the structure is going to be important in its function, okay? And so anyway, you can see some of these sort of different binding domains that are on here, pretty complex type of protein. Okay. Now, here's another one, okay? So ankyrin, okay, is another actin binding protein, okay? Links the spectrin actin network and the plasma membrane, okay? So again, these are all actin binding proteins that are very prominent in those red blood cells, okay? But it's not only in red blood cells, we pretty much want to think about them being in most cells, right? And again, associated with the actin cytoskeleton, so near the periphery of the cell. Okay, So we're going to look at a picture here. Okay, so again, it's telling you that there's another protein that helps link these as well. Okay, so here's your little red blood cell, here's your plasma membrane, right? And if you basically look right at the surface, right, the outside of the cell, again, plasma membrane, outside of the cell, inside the cell. Okay, so what do you see here? You can see actin, right? Actin filaments, and then you can see some of these other proteins that we mentioned that basically help link, right, the cytoskeleton to the membrane and also to these membrane bound proteins or membrane associated proteins. Okay, again, I'm not worried about you memorizing any of this. The idea is to mention some of these actin binding proteins like spectrin and ankyrin that are involved in anchoring that cytoskeleton to the membrane essentially. Okay, they don't bind to the phospholipids, they bind to those proteins, those transmembrane proteins that are in there. Okay. All right, like I said, there are in other cell types, there are similar linking proteins, okay? And so, again, this is what we kind of first found out using red blood cells, but basically you have similar proteins that do the same thing. Sorry, I don't know why this thing keeps popping up. The same thing in other cells as well, okay? So, for instance, dystrophin, and we mentioned dystrophin before, okay? So dystrophin in muscle cells, okay, will link actin filaments to, again, transmembrane proteins, just like I showed you with spectrin there, okay? And then also help link to the extracellular matrix. And this helps maintain cell stability during muscle contraction. So if dystrophin is mutated and it's not functioning properly, you lose the stability, okay? And so when those muscle cells contract, things start pulling away from the plasma membrane and pulling away from the extracellular matrix. Cells start to die, and then you start to lose muscle function. Okay, so again, dystrophin is an actin binding protein. Okay, and so here's more about muscular dystrophy. Okay, we talked about this before. So you get this degeneration of skeletal muscle. Just a reminder that I did post a review article, about dystrophin, and I believe it talks about spectrin and maybe ankyrin as well. Okay. Take-home message is that these proteins that link the cytoskeleton to the plasma membrane and to the extracellular matrix are super important. Okay, again, if you have a misfunction or dysfunction of just one of them, okay, it leads to a very serious disease like muscular dystrophy. Okay, and so if you talk about dystrophin being totally absent, right, you have a more serious form of muscular dystrophy called Duchenne. If it's just abnormal and maybe it has most of its function, it's muscular dystrophy called Becker's. That can be a little more severe or a little less severe. Okay, so again, just kind of a sort of tie-in and a reminder of why it's important to identify these proteins and to study them, right? Okay, now some of these, so we're kind of, I guess we're still talking about actin filaments. That was us talking about some specific actin binding proteins, right? And describing how the actin cytoskeleton is linked to the plasma membrane, okay? And then what happens if one of those proteins is disrupted, like dystrophin, okay? Now, this is, again, kind of talking a little bit more about function of that actin cytoskeleton, okay? And that one other, another function of this cytoskeleton, again, right at the cell periphery, is that it helps form contacts with adjacent cells, right? So I just showed you the picture where you have these actin binding proteins linked to the plasma membrane proteins or transmembrane proteins that then are also linked to the extracellular matrix, okay? Then you can then be linking to another cell, okay? And also too, if you culture cells in a dish, right? We do some cell culture. The cells, certain cells, will actually adhere to the surface of the culture dish. And this all has to do with actin cytoskeleton, and also other proteins, actin binding proteins. Okay? All right, so I'll show you a picture in a second. So it says these regions are, so where they're attaching either to each other, the cells either to each other or to the dish, you'll see it's kind of a site, or something called stress fibers. And we're not really going to talk too much about stress fibers, and don't think that it's like something super different than what we talked about before. But basically, you'll see that there's a lot of actin bundles there, okay, in that area. So let me just show you right here. So this is actually showing you, it's a pretty picture, right? So these are fibroblast cells grown in culture, right? And you can see that actin is stained in purple, right? So you can kind of see the filamentous actin here. All of this is actin, right? Actin cytoskeleton. And then these focal adhesions are stained green. So basically that's where focal adhesions, meaning where it's going to stick to the ditch, right? Okay. And so you can just see that there's a, there you'll kind of see a high localization of actin bundles and stuff like that there. Okay. So it's just kind of showing you that you can sort of label for specific proteins and identify, you know, specific areas. So kind of, like I said, where the green is sort of points of contact with the culture dish. Okay. All right, so that's showing you cells in culture, right, adhering to the dish. I also told you in the last slide that cells will join together with each other, right? If you talk about epithelial cells or epithelial tissue, okay, that, you know, epithelial tissue is formed by a bunch of epithelial cells that join together to kind of form like a sheet, right? And they basically cover things or they line organs, okay? It's super important that those cells are joined together properly, okay? And so, again, this involves actin and the actin cytoskeleton and then other proteins that bind to it, okay? And so that's what this is talking about, cell-to-cell contacts, right? You can call those adherence junctions, again, places where they join together, okay? And again, there are other proteins that are involved in holding these cells together, okay? Some examples are cadherins, okay, and also catenins. Okay, so these are similar to like what we talked about before with the red blood cells, but they're going to provide a specific function in linking cells together, okay? So again, if you look here, so this is showing you here, right? So here's an epithelial cell, here's another epithelial cell, right? They're all lined up next to each other, and again, they have to be joined together to kind of form this sheet-like tissue, right? And so you can see here, this is zooming in on one of these junctions, okay? And if you look at it, you can see here is your two plasma membranes, right? One cell, here's the second cell, okay? And what is this? This is actin filaments, right? The actin cytoskeleton that's really close to the plasma membrane. And then you have a bunch of other actin binding proteins or associated proteins that link the actin cytoskeleton, right, with a transmembrane protein, and then also extend out into the extracellular matrix and then bind to another, essentially another transmembrane protein of another cell, okay? So these guys are linked together, okay? And then these proteins, same thing, link to the actin cytoskeleton of the other cell, right? And so that's how this happens. It's mainly due to the actin cytoskeleton and the associated proteins, okay? So if you guys think about, I'm going to go back to this. So if you think about why this is important, again, right, I told you in epithelial tissue, particularly think about epithelial cells that line your small intestine, right? Or even in your GI tract in general, okay? You have food traveling along in your GI tract over here, right? Eventually that food, it starts to get broken down, right? And then in the small intestine, nutrients will start to get absorbed, right? But there's lots of stuff in the GI tract that we don't want in our bloodstream, right? That the body doesn't want. And that's going to stay in the GI tract and get, excreted out, okay? So the only way the nutrients and, whatever vitamins, minerals can get into your bloodstream, right, is going through these cells, okay? And then they'll end up moving into the bloodstream. If these junctions aren't tight, things can start leaking through, things that shouldn't leak, bacteria, things that you don't want in your bloodstream, right? And you can call that leaky gut, all right? And so again, just a little word on why it's super important that these junctions are maintained properly, okay? All right, now I told you a lot of this is about actin here. So we talked about actin being involved in movement, right? And so how does that occur? So this is first talking about cell surface protrusions. So let's just go back here just to remind you. See these epithelial cells, right? They have, if you're talking about the small intestine, they'll have these folds and then these villi and these microvilli that help increase the surface area, okay? the change in shape in this part of the cell has to do with the actin cytoskeleton, and we would consider this a protrusion, okay? All right, and so anyway, we're going to talk about that, okay? We already basically said this, right? And so here's what I was just talking about before. So again, any cell that has this sort of characteristic shape or some sort of extension, or we're calling it a protrusion here, that's all due to the actin cytoskeleton, okay? All right, so anyway, I already said this, but if you're wondering how much the surface area is actually increased due to these microvilli, it's about 10 or 20-fold. So this is actually showing you an electron micrograph there, right? So that other drawing was not great, right? So this is actually showing you the microvilli, okay, extending. And so what do you think this stuff is, right? This is actin cytoskeleton, right? And then you can see these sort of extending, these protrusions extending up, okay? All has to do with actin in there as well. Okay, so there are other surface protrusions like microvilli that can sort of just happen based on environmental stimuli, okay? Those microvilli are always there and they should always be there, right? Because they're there for absorption. However, there's others that kind of form and then or what have you, okay? So here are some examples of those. So pseudopodia, okay? So these all kind of have different sizes here. So it says a moderate width. If you talk about phagocytosis that I mentioned before, right? A white blood cell engulfing a bacteria, it's going to be pseudopodia that extend, okay? Also, amoeba, the way amoeba move, if you can watch a video on YouTube of amoeba moving around, that's pseudopodia that they extend. Again, all having to do with the actin cytoskeleton. Also, there are extensions or protrusions called lamellipodia, okay? And these are kind of more broad, sheet-like extensions. And that's like when I showed you those fibroblast cells, you could see how those were kind of like, yeah, I mean, exactly like it says, more sheet-like and broad, those extensions, and kind of then when we showed them kind of adhering to the culture dish, right? Those are lamellipodia. Filipodia are thinner projections, okay? And so if you look at a picture here, this is showing you some examples of this. So this is actually showing you, phagocytosis, so a white blood cell engulfing a bacterial cell here, right? And you can see how this stuff, has to extend out to get that, okay? Amoeba showing you it's pseudopodia. Again, these are all electron micrographs. And this is actually showing you lamellipodia here in a tissue culture cell, probably a fibroblast. Okay? All right. Now, we move on from actin. So again, if you kind of think about just, really, if you focus on just actin in terms of its functions, right? It's those cell protrusions, okay? It's cell movement. We talked about, what else did we talk about? Right? Cell protrusions, cell movements, and also that they're isolated or located right at the periphery of the cell, okay? We call it, sort of cortical cytoskeleton. We also talked about joining cells together, right? Those junctions, okay? And so you in epithelial cells, the way those epithelial cells are joined together really involves actin and associated proteins. All right, so we move on to myosin. Okay, so it says myosin is a molecular motor. Okay, so it can convert ATP into mechanical energy and can actually generate force and movement. Okay, if you talk about muscle contraction, it's actin and myosin interacting with each other. Okay, and we're going to talk about that a little bit more. All right, so again, if you're talking about muscle contraction, we're now focusing on muscle cells. Okay, and sorry, it starts off here, right, talking about skeletal muscle. So if you just look at your arm, you look down at your bicep, right, big skeletal muscle, okay? And within that muscle, you have bundles of muscle fibers, okay? Those muscle fibers are single muscle cells, okay? And they're long, they're not round, and most of the cytoplasm of those muscle fibers are composed of something called myofibrils, okay? That are basically myosin and actin filaments. Right? And then each myofibril is basically a chain of units called sarcomeres. Okay? And so that's where you get this striated appearance because of these sarcomeres. Okay? So this is showing you this. Here's your muscle. Here's your, bundle of individual muscle fibers or muscle cells. And you can see that muscle cells are multinucleated. Okay? And you can see that their nuclei are pushed to the edge, right? Because basically the whole cytoplasm of the muscle fiber is composed of these myofibrils, right? And then if you look and just zoom in on one of those myofibrils, you can see sarcomere, right? So here's a sarcomere, here's a sarcomere, here's a sarcomere, just all lined up, okay? And that's what this is showing you here. Those are essentially the functional units of the muscle cell, right? That's where the contraction is actually going to occur. That's where actin and myosin are going to interact with each other. All right, so if you zoom in on the sarcomere, this is what this is, right? An electron micrograph showing you that, and you can see kind of what the sort of, based on the picture, what that corresponds to, okay? And so you notice that there's kind of these different zones and these different bands that are named, and basically it has to do with what proteins exist there, right? And so if you kind of just look here, right, in this area here, it's just what? It's just actin, right? You can only just see actin here, and it looks a lot lighter in color. If you look in this area here, right, you have actin, but you also have myosin there, right? And so you see it's darker because there's more filaments there. And so same thing on the other side, okay? And so we're going to talk a little bit about what happens during muscle contraction, right? So we zoom in here, okay? Again, here's your actin and here's your myosin, okay? And so this is your sarcomere. This whole thing is your sarcomere, okay? And so think about this as it's in its relaxed state. Muscle in its relaxed state. In order for, or when it contracts, the sarcomere shortens, right? So both of these edges pull in, and you notice now, so what you had here is a slight overlap between actin and myosin, right? But at contraction, you basically see that the actin and the myosin are completely overlapped. So they slide essentially past each other, okay? And so that's what this is saying, filament sliding. In actuality, the myosin grabs on really to the actin and kind of pulls it together. Okay, so we can get into more details with this, but we're not. Okay, so just understand that in the sliding filament model of muscle contraction, the actin and the myosin, those are the filaments we're referring to, slide past each other, and you basically get shortening of that sarcomere. Okay, again, it's the myosin grabbing a hold of the actin. I think we may talk about that in a little more detail. So it's not really just simple sliding. Okay, there's a lot more involved, and there's other proteins involved as well. Okay, so that's muscle cells, right? Muscle contraction, muscle cells. You also have sort of contraction occurring in other non-muscle cells. Okay, so the same sort of idea that this sort of actin and myosin or sliding of actin filaments or sliding of filaments to produce contraction happens in other cells as well. So we talked about those stress fibers, right? Where the cells were sort of adhering to the culture dish and where you have sort of adhesion happening. Those are some examples of contractile assemblies as well, okay? And so, again, it's the same idea. It's just that you don't have that really complex sarcomere or anything like that, right? But you have the two filaments here. So again, here's your actin, right? And here's your myosin. And you can see that the myosin, like I told you before, really grabs on to the actin and basically pulls those actin filaments, right? Just pulling this one this way and this one this way, okay? And then again, forming this contraction or the shortening or more overlap, okay? So an example, cytokinesis, right? We talk about mitosis and we go through all the stages of mitosis, and then what happens in cytokinesis, right? That's when the cell is actually pinching into two, right? And it's really like division of the cytoplasm. And during cytokinesis, a contractile ring forms, okay? That kind of, again, squeezes that cell and kind of squeezes it into two, okay? And so that contractile ring has actin and myosin, okay? And so that's what we just, and what's happening there is these actin and myosin filaments sliding. Okay, and so it's showing you a picture here, right? Again, here's your contractile ring. It's shortening and squeezing because those actin filaments and myosin is grabbing onto those actin filaments and pulling them together. Okay, now there are other myosins as well. Okay, there are myosins that are not involved in contraction. So what we just talked about was myosin interacting with actin in a muscle cell, a muscle fiber, to produce contraction. And also in other cells, okay, not just in muscle cells, you also have actin and myosin interacting to produce contraction, okay, like in cytokinesis. There are other myosins that are not involved in contraction, okay, so these are these non-muscle myosins, or I don't know, non-contractile myosins. And so a lot of these function in cell movements and also transport of vesicles and organelles, okay? Yeah, I mean, I would think of it mostly as internal cell transport, right? So moving vesicles around. We talked about the secretory pathway, right? And we said if a protein is destined to be secreted out of the cell, right, it gets made in a ribosome on the ER, enters into the ER, then moves to the Golgi and moves to the surface of the cell. Well, how does it get to the surface of the cell, right? It's transported in a vesicle, but it moves around the cytoskeleton, okay? And with the aid of myosin. Okay, so it's saying basically this is one example. So myosin 1, okay, it will bind to actin and move vesicles around. Okay, so this is an example of, you know, that protein that I just talked about might be packaged in a vesicle, and it will move from the Golgi to the plasma membrane along actin filaments. Okay, or the actin cytoskeleton. And the way that it's able to do that is that you have a particular myosin, this myosin 1, that will bind to actin and also will bind to the vesicle and be able to move it around, okay? The vesicles don't just float to the membrane, they move around, okay? Here's another example, myosin 5. So this one, again, just has a slightly different structure. It's the same idea. It's moving things along actin filaments, okay? This myosin 5 is particularly important in neurons because it's going to be moving around, I guess, neurotransmitters, right? So, and also moving around other things besides just vesicles here, okay? Yeah, all right, so this is just kind of showing you that some of the other ones are involved in actin reorganization or anchoring. So we kind of talked about, don't worry about this. I think you're going to get, let's just stick with, we're talking about myosins moving things along the actin filaments, okay? And so, again, if you think about neurons, they a lot of times will make the neurotransmitter in the cell body, right? And then that neurotransmitter has to be transported all the way from the cell body all the way down to the axon terminal. And so super important that myosin is functioning properly and is able to move these things along the actin cytoskeleton, okay? And so that's why it's mentioning that, because it sort of has a longer distance to go. So if the neurotransmitter doesn't get to the axon terminal and isn't able to be released, then it's not going to stimulate the other cell, right? And so then those two neurons wouldn't be able to talk to each other. Okay, so this is kind of, again, this is just showing you the same idea. It's just that this myosin has a different structure, and what it might grab onto might be slightly different than just a vesicle, okay? But it's the same idea. It just finds cargo on this side. I can move it around. This is actually showing you that it's grabbing a hold onto an intermediate filament, right? We said intermediate filaments are also part of the cytoskeleton, so you can kind of see how this is sort of linking the cytoskeleton, kind of linking three components together, right? Here's your actin, myosin, and intermediate filament. But bottom line is this can grab onto a lot of things, okay? And again, it's going to move things along the actin filaments. Okay. So if we talk about, finally move away from actin and myosin and talk about microtubules. So I think what I'm going to do is I'm going to stop here and I'm going to upload this, and then I will pick up with, I guess it doesn't really matter. Yeah, let me do that. I'm going to stop here, okay? And then I will upload a second one with the last half of it, because I know this is a lot. So at least you can kind of stop here, regroup. So we will start, I'll put another one up with microtubules. So I'll just label this one as cytoskeleton 1, and then I'll label the next one as cytoskeleton 2. Okay?